Hello everyone, I'm Omkar. Today we are going to see how to target and cure hepatitis B using CRISPR-Cas9. I have taken up this project as a part of my course Elements of Synthetic Biology at Dhirubhai Ambani Institute of Information and Communication Technology. So, what is hepatitis? Hepatitis is a viral infection that attacks the liver. Hepatitis can be caused by the virus, drugs, or alcohol, although the most common factor is virus and is known as viral hepatitis. There are several types of viral hepatitis. Amongst them, the most common are hepatitis A, hepatitis B, and hepatitis C, each differing in more than 8% of the nucleotides. Hepatitis can manifest either as an acute or as a chronic disease depending upon the causes. Now, let us see what hepatitis B is. Hepatitis B is a severe form of viral hepatitis transmitted in infected blood, commonly affecting hepatocytes, the liver cells. It can be seen in both adults and infants. The person affected with this infection may have symptoms, or sometimes the symptoms might not be visible. A clear picture of hepatitis virus can be seen in the image. So, how do we test for hepatitis B? A simple blood test can show whether you have hepatitis B. The blood sample is collected from your liver and is taken using a sterilized needle. Presence of hepatitis B vaccination in your body can also be found out from this blood test. Coming to the symptoms, there are various symptoms like feeling very tired, mild fever with headache, loss of appetite, feeling sick to your stomach or vomiting, stomachache, dark urine, jaundice. Jaundice usually appears only after other symptoms have started to go away. Symptoms appear about 3 months after the contact with the virus. Causes. There are various causes like having sex with an infected person without using a condom, sharing needles used for injecting drugs with an infected person, get a tattoo or piercing with tools that weren't sterilized, share personal items like razors or toothbrushes with an infected person, to the infants during delivery. A mother who has the virus can pass it to her baby during delivery. Medical experts recommend that all pregnant women get tested for hepatitis B. If you have the virus, your baby can get shots to help prevent infection with the virus. One cannot get hepatitis B from casual contact such as hugging, kissing, sneezing, coughing, or sharing food or drinks. In major cases, hepatitis B goes away on its own. A healthy diet, adequate rest, drinking plenty of water, and following doctor's prescription can relieve you from the infection. So, as the saying goes, prevention is always better than cure. Here are some prevention methods. The prevention methods are like using a condom when you have sex, not sharing needles with many people or sterilizing them regularly, wear latex or plastic gloves if you have to touch blood, avoid sharing toothbrushes or razors, don't get a tattoo, or make sure that the needles used have been cleaned properly and are sterile. The best prevention method is vaccination. The hepatitis B vaccine is made from a synthetic yeast. The side effects of the vaccine are redness and soreness on the arm where the shot is given just for a short period of time. Now, let us see what chronic hepatitis B is. The advanced stage of hepatitis B is chronic hepatitis B. It is majorly seen in children and young people who are affected with hepatitis B. 90% of the infants won't be able to get rid of the virus and develop chronic hepatitis B. People infected with chronic hepatitis B have no symptoms. Treatment for chronic hepatitis B depends on the level of infection in your liver and if it is increasing or stable. So, what happens when, how does the virus enter our body, or how does it infect the liver? When the virus infects the human body, the outer shell, the yellow shell, that breaks away, and all that is left is the caspid and the virus inside it. Caspid is the protein shell of the virus. The caspid, along with the virus, migrates to the liver cells and delivers the genomic rcDNA, relaxed circular DNA, which is then converted into the cccDNA, covalently closed circular DNA. It is a special structured DNA that is formed during the migration of the virus cells into the nucleus of the host cells. This cccDNA attacks the genome of the host cells and fuses itself into the host DNA. So, how do we cure chronic hepatitis B? There are several treatments available, and CRISPR-Cas9 is considered most efficient till now. Though research is still going on, it is worth shedding some light on it. CRISPR, clustered regularly interspaced short palindromic repeats. CRISPR is a defense mechanism which occurs naturally. It is found in a wide range of bacteria in the nature. CRISPR is one of the part of bacteria's immune system. CRISPR can keep track of the bits of dangerous viruses around itself like an image, such that it can recognize them and defend against them next time whenever they attack. These bits guide the Cas9 to a specific portion of the DNA where the virus has attacked. The defense mechanism's second part includes some set of enzymes called CRISPR associated Cas proteins. The image shows how Cas9 works. As you can see from this image, this Cas9 can precisely snip the DNA and cut them with the tiny molecular scissors. Conveniently, the genes that encode for Cas, they are always sitting somewhere near the CRISPR sequences so that they can defend against the dangerous viruses. From the adaptive immune system of the bacteria, this whole CRISPR-Cas9 system is derived. Targeting hepatitis B with CRISPR. Human body does not have natural CRISPR mechanism. We try to fuse this mechanism as a treatment into the human body. To do that, first we need to find a guide RNA for the Cas9 to guide it and cut the virus DNA. For cutting the cccDNA from the hepatocytes, we need to find a guide RNA that will guide us to the target sites with less off-target effects. For this, we need to study the genome of hepatitis B virus. As you can see from the image, the schematic representation of the hepatitis B genome, the open ring frames core C, polymerase P, surface S and X proteins are the viral transcripts. The dotted line represents the cccDNA of the genome and the thin line represents the rcDNA of the genome. As we can see, some parts of the ORFS and ORFX converge the cccDNA, and these are the conservation regions. These conservation sequences form the single guide RNA, and the protospacer adjacent motifs, that is PAMs, were identified in this sequence itself. These are the target sequences. It is the DNA sequence and sequence conservation of the regions targeted by Cas9 within the S and X gene of hepatitis B virus. Target sequence in ORFS and X are depicted as S1 and S2 and X1 and X2. Each of them have protospacer adjacent motifs, PAMs, represented by the bold box, as you can see it from the image. The boxes show two pair of 20 nucleotides complementary to gRNA, and offset distance between the two sequences complementary to gRNA is underlined. Conclusion. Above analysis in CRISPR-Cas9 systems directly target an authentic pathogenic virus and demonstrate the potential for cccDNA directed antiviral therapy using Cas9, which may represent a significant step towards the cure of chronic hepatitis B virus infection. There is a risk of mutation after cutting the viral DNA due to off-target effects. Recent developments and